Thank you very much. So first of all, I want to thank to be invited, and also I would like to thank you all for the nice previous presentations. And I really wish I was there to be able to discuss and be part of this. Anyhow, I will try to tell you a little bit about the work we have been doing when it comes to sterile fish used for aquaculture. And just my background is that I'm a scientist at the Institute of Marine Research in Norway, where one of our tasks is then to assay the effects of aquaculture on the Norwegian coast, and in this context to solve some of the sustainability problems that are occurring. So most salmon aquaculture is based on the coast, and the salmon is kept in open net pens like this. And this causes quite a lot of sustainability problems. For example, you have the salmon lice problem, which is a disease. Also, the feed is a problem. And then you have welfare issues. And then, of course, these net pens can break and you can get escapees and you have wild salmon swimming by. And we see solutions with genetic engineering that we could use this to introduce disease resistant or maybe convert the fish so that you can feed them on sustainable feeds. So it's becoming even more important to solve and use sterile fish. So this is just an illustration showing how much farmed fish you find on the coast of Norway. And you see this tiny bar to the right. That's the wild fish swimming by trying to breed. So one of these containers can contain up to 200,000 fish. So if they escape, they really can have a large impact on the genetic integrity of the wild fish, which is not a large problem maybe now, but in a few years, as more genomic selection is coming and also use of gene editing, this can become a larger problem. So currently, sterile salmon is mainly triploids in the industry, and also this is you can make triploids. It's sort of a genetic engineering the old way because all the salmon eggs are diploid upon fertilization, and right after fertilization the egg is triploid. So by pressure treating the eggs, the embryo becomes triploid and so forth sterile. However, this comes with a lot of drawbacks because they're very sensitive to being in the sea cage since you have a much more environmental changes. You will have much more skeletal deformity, cataract formation, and also the males still go through puberty, so they will compete with the wild males on the breeding ground. And they're also not always 100% sterile. So in my group, we've been working with alternative to solutions for making sterile fish, and that includes mainly going for two stages what I showed here. Either you inhibit germ cell formation by targeted protease essential for the primordial germ cell formation, or you try to prevent maturation when the fish is then vaccinated, which is about the small stage. Or what you also can do is you make a knockout and then you try to induce fertility at this 3 stage. I'm not going to show anything from that, but basically there have been people working on all sort of methodologies to try to make the fish sterile, in which vaccination has been tried out, also knockdown studies, which are sort of working a little bit. The problem is that you don't get 100% using these technologies. And we have then started to use gene editing to make germ cell-free fish and also immature fish. I'm coming back to that. And then in the end, by combining different knockout models, this Japanese group, Goro Sasaki, has created actually a very nice method where you can go surrogacy by transplanting germ cells and then mass producing sterile fish, which I'm not coming into, but this is maybe the future. So anyway, just show you a little bit how it's done in salmon. While you CRISPR the fish at the one cell stage by injecting the CRISPR, and then it's very efficient. You get very high mutation rate already in the F0. So here we target albino gene, and you see you get very high mutations rate above both alleles. And we also have developed this method further now, where we can use HDR then to induce the types of mutation we would like at the target site. But the mutants we are working with right now in the second generation, they're all on a more used with the old technology. So one of the things that we have is that we have a great mosaicism in the F0 generation, where we also can have GNR that creates a total knockouts in the F0, which I'm going to show you, but also where we have to wait to the F1 generation to create the knockout because we have a lot of inframe mutations. But basically, this is how we've been working. I just wanted to illustrate this because when you do a normal cross-out in a model species or something, it takes a very long time to do this in salmon. So instead of we make the F0, but then we cross the F0s out to each other to make the F1 to be able to create knockouts already in the second generation, because the generation time is 3 years. So basically what I'm going to show you today is mutants that we made that are F0 and also F1s, because of in the F0 we had too much inflammation, so we had to go to the F1. So just wanted to show you how it can be done. This is just how, when we have the mature fish, and sort of not always so easy to strip them at this stage. So coming back to making germ cell-free fish, we have now knocked out 2 genes. Actually, we just published a paper this week where we knock out the PV1 gene, but we previously knocked out the dead-end gene. Both of these mutations are then involved, or these proteins are involved in primordial germ cell formation. So if you remove these genes, you lose the germ cells. Just illustrated here with the picture to the right, where you have a knockout fish without oocytes in the ovaries. Also, we have then followed this fish to measure both the welfare and the production phenotypes and the meat quality. And you can read about this. I'm not going to talk too much about this now, but we have made sort of studies that tells us that these fish are very suitable for production. They don't enter puberty, so you don't have welfare problems with puberty. They also have indistinguishable welfare when it compares to wild type, and also we measure the meat quality, and they actually have sort of a nice feature in the meat quality is that they actually contain relatively more omega-3 in the whole gross fat than a wild type fish. So, but the problem when you come to this is that, of course, you can't breed them to the next generation. This is what you want to do when you want to mass produce sterile fish. So we've been working with the model where we're trying to increase or try to get over this hurdle that they're germ cell free by then rescuing the knockout. So first we knock out the dead gene, but at the same time add the mRNA so that we can get germ cells in the next generation, and this is what we call the virgin broodstock. So it's basically genetically sterile fish, but still they have germ cells. And just showing how it can look like. So we have here knocked out the dead gene plus the albino gene, but then we also added the mRNA, so we get oocytes formed in this fish, and the same for the males. And now in the latest few years that we've been working with getting these fish to mature, and now we're actually working on the F1 generation, and there we have a lot of dead and knockout fish and a lot of sterile fish, because every female can produce up to 8,000 eggs. So we have at least 2,000 or 3,000 sterile offspring that we currently are going to apply for a research license to put them in the sea to see how they're doing in a real sea cave situation. And also we have then tried to create a new generation by adding the dead end mRNA to this fish so that we get fertile, sterile, genetically sterile fish again. And this is current work that I'm not going to tell you too much about, but we hope to be able to publish this in the coming year. So another feature that we worked with is welfare, and one of the welfare problems is puberty, precocious maturity, and this is a problem in most aquaculture species, because when you feed the fish a lot, the males tend to mature, and this also stops the growth of the males, but it also increases disease susceptibility. And you get more disease in the facility, plus that you get not so good meat quality. And this is until now it's solved by using continuous light to delay maturation, and also genetics delay maturation allele. However, with increased use of online facilities and also higher temperature seas, still the fish mature. So we have been working with the maturation, and in this case, I'm just not going to tell you which gene it is, but we worked with one gene that's involved in maturation, and we knocked this gene out in the F0, and we crossed these F0s out to create the F1 generation. In the F0, we didn't see much phenotype of knocking out the gene in terms of maturation, but when we came to the F1, we have a very clear phenotype, and we believe this is due to we have a lot of infrared mutation in the F0. So the F1 fish of this, what we now call the forever young gene, was that it seems like, at least we looked into the males, and the males seem to be not maturing at all. They stay immature, or they are forever young, while all the males that we assayed in this study did mature, and they have all stages of germ cell or sperm production. And also when you look at the GSI, both 7 and 11 months after you had induction maturation in males, you can see that all the knockout fish, they do not enter maturation. They have a very little size of the gonad. This is the yellow bars, while all the controls mature. But interesting, I think this is also something that's interesting in this fora is the heterozygous fish. They show what we call an intermediate phenotype, meaning that we had some fish that did mature and something that didn't mature. And I think this is a very interesting phenotype, because you can control maturation also by using environmental triggers. So I think using this heterozygous fish can be very interesting for farmers, just to maybe reduce the amount of maturation, and also using the right conditions, this dose dependence of this gene. You can actually get much less maturation without using very exhaustive environmental triggers. So in the end, I just want to acknowledge the people in my group that's been doing a lot of this work, plus collaborators from Embrapa, Fernanda Almeida, and my former postdoc Hilal, who's done a lot of work with the dead end knockout, and is now working at the university in Czechia. So thank you very much.